Buonasera, buonasera a tutti. Benvenuti a
Good evening. Welcome to GiovedìScienza Science on a Thursday. We will be talking about plants today, but plants that are not an island. In fact, they are the queens of cooperation and interaction with microorganisms, and we will soon be seeing how many and what type of microorganisms. There are lots and lots of them, and we have one of the greatest experts of interactions between plants and microorganisms, Paola Bonfante. She is a member of the Academy of Science of Turin, of the Accademia dei Lincei in Rome. She is one of the most quoted. She is an emeritus from the University of Turin. The other guest is sitting at my right, Renato Bruni from the University of Parma, where he is the director of the botanical gardens. As well as being a scientist and a lecturer, he is also a popularizer, very good and greatly appreciated. He has a blog which is called Erba Volant that you can find very easily. He has authored a number of books, and I would mention the one which refers to today, which is called Le piante son brutte bestie, plants are difficult animals, and published by Codice, and you will find it in a bookshop or you can order it online.
Piero Bianucci.
Ma io non ho granché da dire.
And obviously, we have our Piero Bianucci. I haven't got much to say, except that I was thinking on the title of this meeting. In fact, there are more microorganisms on Earth than there are stars in the skies. And this is absolutely true, because the number of stars would appear to be enormous, unlimited, but in fact you can estimate it quite accurately. Usually, we say that there are about 100 billion stars for each galaxy, and so our Milky Way is an object made up of 100 billion stars, possibly 300 billion, but ours is quite big. On average, galaxies are smaller than the Milky Way, so let us say 100 billion stars per galaxy. How many galaxies there are, this is more uncertain, but we think another 100 billion ones, so altogether they are 100 billion by 100 billion. This is 10 to the power of 22 number of stars, a number which is not easily imagined or envisaged, but certainly much smaller than the number of molecules that there are in 18 grams of water. So a very big number for us to comprehend, but not such a big number when, in fact, you go to see how many zeros there are. And there are lots of zeros with microorganisms of plants, more than the stars. Yes, again, they're estimates, because no one has gone there and counted them one by one. It's not so easy, but one of the estimates, which refers to microorganisms that live on the surface, on the outside of plants, is 10 to the power of 26, 10 to the 26. So don't ask me to transform this into billions of millions, but it's bigger than what was said before. It's a finite number, but it's big. And we will see this evening that there are not only the microorganisms on the surface of leaves or plants, but the ones that are inside. So if we wanted to count them all, this number is very, very big. So just on the surface of plants, we have 10,000 times more elements than there are stars. This is quite a stunning beginning. So a couple of billion per cubic centimeter. If we want to switch it round, imagine how many interactions or how high the number of possible interactions can be. So it can be very complex to understand where these interactions go to, how you can read them. You must be very good at maths. We will enter the conference, and we will do so with a cartoon.
Microbiomes are collections of microorganisms that live together in a given environment. Every soil and sediment on Earth has a microbiome. There are microbiomes in the ocean and even in the atmosphere. In your body, there are microbiomes in your skin, in your mouth, your digestive tract, anywhere the body comes into contact with the outside world. All life on Earth evolved together, so microbes need us and we need them. We depend on microorganisms to help us break down nutrients and produce important vitamins and enzymes in our digestive system. It's estimated that there are over 1,000 different kinds of microorganisms living inside our bodies, containing about 300 times more genes than our own human cells. If you put together all the microorganisms inside you into one mass, it would weigh as much as 6 pounds. In comparison, your brain only weighs about 3 pounds. Each of us carries inside of us a unique fingerprint of microbial diversity. Your unique microbiome starts as soon as you're born and is influenced by your diet, your experiences, what you're exposed to, and where you live. In some cases, your microbiomes can even affect your mood and behavior. Scientists believe there are several pathways through which the bacteria and other microbes in your body influence your brain chemistry. For example, experiments have shown that organisms that are raised in a completely sterile environment perform differently in tasks related to learning, memory, and social behavior. Currently, scientists don't have a perfect understanding of how these microbiomes work. Although we can catalog with incredible precision and speed the number of microbes living inside of us and around us, we still have a large gap in knowing how these communities interact with each other and with their environments. To develop the kinds of tools needed to study microbial communities and how they function, we need a unified approach that draws on many different disciplines. From the inside of a single cell up to the size of our entire planet, we need new technologies and methods to help us decipher the working principles of these communities. If we can understand these complex systems, it would have a large impact on how we interact with the natural world. There are applications in agriculture and crop productivity, in climate science and carbon cycling, in sustainable energy and biofuels, and in health and medicine to treat disease or create new therapies. Understanding microbiomes may even one day help us to colonize new planets. Microbes were here on Earth before us, and they're an important part of what makes this planet habitable. If we wanted to colonize Mars, for example, it would be critical to understand the role microbes play and how they integrate with a new ecosystem. We're only just now beginning to understand the full breadth of services microbes have to offer. There's a whole invisible microbial world out there that we didn't know existed before, and we're almost at the cusp of understanding this huge part of our environment and our own bodies. Microorganism communities may be small, but they're also very important. As they say, it's the little things that make a big difference.
Paola Bonfante. Dunque, sono le cose piccole a fare una grande
Paola
differenza.
Bonfante.
Capire qualcosa di
It's
più
the small
fondamentale
things that make a big difference. Should we try to understand and comment what we've just seen?
Per dire che effettivamente questi piccoli microrganismi
I must say that this is an opportunity to say that, yes, these very small microorganisms that we saw in this rather fast but happy cartoon shows us it gives us an idea of a great revolution of biology. That is to say, if we look at these microorganisms, just as we heard in the cartoon, we will appreciate immediately that the microorganisms are a second genome or something like that. And what has happened is that many aspects of biomedicine have been affected by it. Sometimes there are jokes that you see, so young researchers or young biomedical researchers must say, if we want to be visible, we certainly have to study this second, in quotes, genome. Now, it's also useful to do something which might be a bit boring, but which is to give a few definitions, because in the cartoon we heard some different words being used, and certainly there are two that are recurring. One is microbiota and the other one is microbiome. Very often these two words are used in an inaccurate manner because the meaning is very different. When we say microbiota, we're thinking of biodiversity of microorganisms. For example, if you look at it in the small image, you see lots of different colored microorganisms that have very different structures and surfaces. different pili or flagellae. And what does this do? It gives us an idea of the biodiversity which exists in a microbial community. So microbiota tells us who's in there, while a microbiome refers to the fact that that particular number of microorganisms has its own genetic information, and therefore the genetic information that is present in it gives us an idea of what it is. This answers another question, that is to say, what type of potential all this community has.
I microorganismi e possiamo vedere.
Well, look at the microbiota. We see the microorganisms and we see who they are, what these microorganisms are. When we look at the microbiome, that is to say, analyzing them in more detail, we can discover what they do exactly, or how we could exploit them. And this is the meaning, because with the microbiome, clearly we reach the genome of the single bacterium that is present in the microbiota. So this idea of the microbiome is linked to another word which is greatly used and which will reoccur probably this evening, which is the metagenome, that all the organisms whose genetic makeup is identifiable when you're studying a niche. This gives us an idea of the range of complexity which we might have when we're talking about microorganisms. Can I ask a question, please? In the microbiota, so far we've spoken about microorganisms in a generic term, but in fact they're bacteria, but there are also, I believe, also fungi or viruses. The population is varied. Thank you. Yes, Dr. Bianucci, for this question. Yes. In fact, sometimes we say microorganisms, but what do we mean by the word organism? This is a sort of umbrella definition that includes many different organisms, viruses, fungi, protists. That is, the idea is that these are something that we can't see with the naked eye. And on the whole, we talk about microorganisms when we talk about unicellular organisms, but I would like to say that there are exceptions. For example, coronavirus is a virus, and as such, we know that it is an entity that lives and multiplies within a cell, but it is not per se a microorganism. Or we talk about fungi. Fungi are very important. Not all fungi, however, are below the threshold of visibility. For example, here is a truffle, and I think this is half a kilo. Certainly, this is not a microorganism. You can see it with the naked eye. Just to say that in definitions, then clearly there are a series of difficulties to fit organisms in those definitions. This idea of a definition which is difficult to give because the boundary is very difficult is another topic which will be recurring. Yes, because it opens a number of more theoretical approaches, and this is a point. Now, going back to one of the key points that was raised in the cartoon, we used to say that in the human body there were very many microorganisms, and in fact, for years, roughly since 2010 to the present, we were used to saying that in the human body there were about 10 cells for each human cell. But recent data published recently show that the ratio is probably 1:1, but this doesn't really change the situation. But it tells us that those counts, as a colleague was saying earlier on, are never so easy or so clear-cut. They have to be verified and corrected. So we have this second genome, and this means that we have to reflect, as can be seen in these images, on the fact that we are formed by human cells, but we have as many bacterial cells. So The question is, what is the origin of this microbiota? And this is also very interesting in terms of evolution. Now, let us move on to plants, because the main topic is plants. The microorganisms are very plentiful, but where are they in plants? Yes, well, we can map the various communities that there are in plants. And first of all, we have a metaphor, that is to say that no plant is an island, which I've been using for a while, referring to John Donne's poem, because the idea is that, like humans, plants do not live alone. They live in interaction with other plants, and this in itself is a first point, but also with all these microorganisms that we listed earlier on. And for this reason, we can map the plants and microorganisms and see where the microbiota develops. So let us start now from above and from outside. Yes, we mentioned this before, that enormous number of microorganisms, which we believe lives on the surface of plants. The number is a number of cells, it's not of different species. So that's what we were calculating, bacteria, cells, or fungi, and so on. We tend to look at ourselves. At a certain point, we realized that what we were starting to discover on ourselves, that is to say, our interaction with microorganisms, was also present in plants. And we started to discover that plants have on their outer surface a number of species. So the number of species on the surface of leaves, that what is called phytosphere, is smaller than other parts of the plant, because it is an area which is a little difficult for the microorganism, because leaves are exposed to light, so there are fewer nutrients. Exposure to light is stronger, not as much water available. These are populations that have developed their own niche to survive. But this large number, in fact, raises a question. That is to say, what are they there for and why? So let us ask this question. Yes. So those microorganisms are there prevalently because plants allow them to stay there. In fact, on plants, there are structures or microecologies or microenvironments that favor some microorganisms and, in fact, select them and deselect the microorganisms they don't want. For example, on the surface of leaves, there is a sort of, what can I use as a word? If I want to use a metaphor, it's a sort of dissuader. That is to say, there are nanostructures, which are like the ones that you use to keep birds away, but there are micro or structures that favor some organisms and hinder others. And this is not a stable situation or not a standard one. No, it isn't, because we have to talk about the surface and within the plants and within the roots of a dynamic situation whereby plants favor some microorganisms and some microorganisms take advantage of the hospitality of the plants, and all this changes. For instance, according also to the moments of the year. I was talking about leaves, but the microorganisms that are on the surface of leaves are different from the ones that we find on flowers, for instance, because in that case the ecosystem changes and therefore the conditions change. One of the main functions of these microorganisms, the ones on the outer surfaces of leaves, is to create a competition of the disadvantaged, that is for the plants, that is to say the organisms that would disadvantage the plant. And the plants, for example, might fear flood or a strong rain, a shower, because this might get rid of the favorable microorganisms and, in fact, allow for the less favorable ones to develop. I'll give you an example. When there is rain, the first drops of rain, the plant has a defensive reaction before the microorganisms that are noxious may arrive. It's a sort of danger signal, and that means that the plant has a sort of polarization of reactions, because all the things that are favorable could also turn into a disadvantage. Things may switch and turn round according to conditions. Can I venture to say that this is very much like what happens on human skin? No, this is what we do, in fact. In fact, there is a balance, an equilibrium on our skin, and that may change. In fact, this also raises another point. How do these microorganisms develop or go there? Rains is a system whereby they can get onto the surface of the plant from the ground. All these drops, because the drops are the ones that we fear, because our saliva might transmit it, may, if there's a strong rain, carry the microorganisms elsewhere. This, in fact, is the way in which microorganisms colonize the surface. Now, continuing with the mapping, the geography, so to speak, we go inside. Inside, we have a local story. Local. And this is Turin's story. At the beginning of the Susa Valley, there is an abbey, the Abbey of Sant'Antonino di Ranverso. Now, lucky, we have solved the problem, but it was famous because people who suffered from ergotism were treated. For example, if they were eating rye, that is or ergots, developed some symptoms. Had delirium, but at low doses they caused gangrene. So there were problems with the upper and lower limbs. At a certain point, especially in the US, they discovered that some animals that were grazing suffered of the same problem. So they were starting to have cases, and in fact, the loss of a limb or severe problems of gangrene, which were very similar to the ones that many centuries ago had been treated in the Susa Valley. What we discovered is that these animals did not eat contaminated rye by the same fungi. They, in fact, ate a plant called festuca, and in particular they were eating the one that grew very rapidly, and only a few years ago we discovered that it generated the same ergot alkaloids, which our ancestors in the medieval times had suffered from. It's not a contaminant, and it doesn't produce the substance, but there are fungi that grow within this ergot alkaloid that has not a symbiotic relationship, but they're just hosted, partly tolerated by the plant, that grow within and generate this toxin. Now, clearly, for this plant, it is an advantage because it means that it can defend itself from grazing animals, from herbivores, and so it has contracted out, so to speak, part of its defense system. These microorganisms live inside the plant without bothering it, and they're an example of what we call endophyte fungi. And until a few years ago, we did not suspect the existence of them because there's a very important new avenue in what I'm saying is that many microorganisms was something that we couldn't see. because it was difficult for us to get them to grow outside the plant. So we weren't observing them, we didn't know that they existed. For example, we went to see, we analyzed the genetic study of plants, and we discovered that there was some DNA we didn't know about. So in cases like this, these cases are in fact increased, not just with the poisoning of people or plants, but we discovered that many plants have endophytic populations that are already in the seeds in some cases. So when the plant germinates, they're already present. They're transmitted from parent plant to the next generation, and they determine the conditions which will favor the plant. The plant will grow more vigorously, the substance will be useful for the plant as a defensive one, and in some cases they will also produce substances that are very important. There's an anti-cancer drug which is called Taxol, or paclitaxel, which we cannot synthesize alone or in a lab. It's one of the cases of a natural drug. It's used in a hospital environment, and that we get from the plant, which is a Pacific yew or Taxus brevifolia. And to treat all these people, we would have to destroy the plants. But we discovered that this plant has a microorganism that is able to produce this molecule. And so what we're trying to do is that we're trying to get the fungus to produce it outside the plant. But unfortunately, it is a bit reluctant to do so. Technology enables us to do this, but it also makes it possible for us to develop this field. I'm not going to make the joke on this field of study, and we will see it in a few minutes. We will see how studies started a long time ago, but in the past 20, 25 years, or more recently, they have increased dramatically. But before we do this, let's continue to map these plants.
Che non sospettavamo ci fosse.
Soil is something we didn't suspect. Yes, I agree. In fact, the great reserve of microorganisms of all the plant is the soil, earth. So yesterday I went to the botanical garden. That was below a rhododendron. So we know that the idea that one time mothers used to say, don't play with earth, with soil, it's dangerous. In fact, this soil, this earth, this black one is very rich. And so this idea there is this exchange between soil and the plant through the root. So a root is the point of contact, the interface. So the idea is, how can we do it? How can we understand what there is in the soil? How can we shed light on this and therefore manage these billions of microorganisms which exist and give them a name? We have no doubts that we need the technology, which is, for example, the DNA-based one, and we can talk about this in more detail if you're interested in it. But thanks to the idea of the fact that you can extract DNA, we no longer need to cultivate microorganisms, and therefore we can immediately analyze them according to their genetic sequence. And we can see, as you will observe in this slide, that plants, as we were saying, is not an island, but it is surrounded by these microorganisms that are very diverse. There are algae, there are nematoids, algae, fungi, every protozoa. We can name them. And if you can see this slide, you can also see that we can identify not only the name, but the number per gram of soil all these microorganisms are. So naming them is very important, actinobacteria, protobacteria, but the interesting thing is that on the whole, the microorganisms that are present there are much more numerous than the ones which enter the root. So there is already a very active selective mechanism. This is one of the most interesting processes. How does this process take place? How does the plant select some of all these varieties, of these taxonomic varieties in the soil? So this, for instance, is very useful because it makes it possible for us to compare these microorganisms in plants with what happens in human beings. Now, please. Yes, well, all right, then I'll go on. Yes, this slide is a little bit difficult, but what does it show us? It shows us, going back to the notion of geography, which we were talking about before, this is something we can compare, enables us to compare the organisms of the plant, the microorganisms of the plant, to the human body. That is to say, our digestive system. So what we see is that it is exactly like the human body, where we know that most of the microorganisms are present in the terminal part of our digestive system, i.e. in the colon. And likewise, in the plants, most microorganisms are in the areas very close to the roots. And then there is the selection-deselection system. But what I'm always very curious about, and has been said by Professor Bruni before, is that these communities of microorganisms are very diverse. So one of the first reasons is why? Because the microorganisms that are in the human body, and you can see in blue here, is that they are basically anaerobic, that become progressively more abundant as we move towards the colon, while in the plant they're all aerobic with very few. Could you please specify the difference? Well, an anaerobic organism can live without oxygen, while the aerobic one is the one that absolutely needs oxygen. And then we'll see that there are some exceptions for plants, but roughly that's the difference. So you can see in those color bands that they change. That is the reason why there is no direct correspondence. But if we then go and look at the functions, and this is really very interesting, functions are very, very similar and comparable, both in plants and in humans. Basically, microorganisms help the host to improve nutrition, to modulate the process, the morphogenetic processes, but above all as a defense against pathogens. So what was mentioned before can be further developed. Plants, like human beings, have to decide how they're going to behave as in relation to what arrives. So how does it decide how and which microorganisms can be incorporated and which cannot, which are part of me, so to speak, and which are other than me? So in the development of plant immunity has recently been flourishing, blooming. So I think it's very interesting to see how there are some resemblances, or there's some resemblance between plants and human beings. Yes, there is great resemblance also in the way in which it develops. Our flora develops in our gut. Our gut flora develops according to the food we eat and to our age and history and condition. For example, people who live in Florence have a completely different gut flora to those who live in Timbuktu or who might live in China. And in the case, for example, of plants, once again, the life of the single plant or the environment in which the plant lives in can change the type of microbiota. And this has an impact also and is very important for farming. That is to say, the same plant in different places can recruit different microorganisms, or it can, in some cases, recruit or manage differently its microorganisms. So different microorganic environments. So this is why we want to study the reasons why there are certain microorganisms and why not, what type of phenomena there are. This forces us, in fact, to do something which is very difficult to understand, but could I use the word holistic? We need a holistic view. We have to see the plant framed in its environment and with all the interactions that it has. The aim is not just to study a plant in detail. We were saying earlier on that we know that we have these microorganisms that have a relationship with plants. We have known this for quite a long time, but only recently we have increased our knowledge. And going back to the parallel with the stars and astronomy, you at one point you pointed your telescope to the sky or down to the plants. In 1974, Paola Bonfante looked at some fungi and discovered something very particular within there. And here it leads us down another path. But only a few years later was it possible to understand exactly what they were and how they behaved. Yes, thank you very much to remind us all of the story of, well, I would like to add that the very young Paola Bonfante, the recent graduate, looked in a microscope and saw that there was a bacterium in a fungus and asked herself why. Now, all this became meaningful with the famous development of science, so the discovery of PCR and the possibility of naming the microorganism there, which at the time could not be cultivated. And this is the notion which underlies our approach on microbiota as we're discussing it today, because it is thanks to a number of techniques that are based on the omic system are we able to see it. We also have a lot of data banks, and all this type of work is basically done by IT specialists. We now know that we can extract these sequences, and as a result, we can name the microorganism that we can see in the e-microscope or electronic microscope. We can just look at the DNA if we want to. What does this lead us to say? Well, it leads us to say that with these techniques, that are so-called omics technologies, to describe the presence of microorganisms in various substances. Water, for example. I would like to remind you about Vente, who was one of the first who used the omics technologies to describe the microorganisms in oceans. So what does this make it possible for us to do? It makes it possible for us to say that we don't only study the microorganisms or microbes that can be grown. We used to put these on a dish, we used to and then grow them. And here you can see an example of it. If you put them on a dish, you have a number of bacteria. But it's not that this is not important. Clearly, we have to develop this notion, which is called of culturomics, or growing them. But first and foremost, we have to analyze them to gain a better understanding of what is in them. This has been a very important, a key tool, helping us to discover a number of new microorganisms, identifying them on the basis of their genome. This made it possible also for us to identify the potential and very interesting molecules. For example, the new antibiotics are based, for example, of this new technology. And the last point, which I would like to stress, because I find it very amusing, is to say all this big omics work. What does it entail for a biologist, the one with a white coat? Well, the work has become much more limited. That is to say, basically, you have to design the project to see how you will take the sample. But once you've extracted the DNA and you've sent it for sequencing, then it's all IT job. We do it at the computer, and at that point you can do it in remote, and so I think the approach has changed. The experimental approach has really changed. So it's a completely different perspective. And at this point, we have other disciplines that join in, because something very similar happens. So when we try to discover not only how many microorganisms there are, but we want to understand what interactions they have with the host. And so in this case, there are other disciplines which are linked to the omics technology, which make it possible for us to study how the changing of a microbial population changes not just the production of one or two molecules within the plant, but in fact we can now look at all the molecules of the plant. We can go and observe, once again in a holistic, comprehensive approach, the metabolic biochemical response that this plant has, and see what happens to its microorganisms. Here we have moved from, can I tell you about a story? Well, here it's a fairy tale, and there are 6 blind or visually impaired people and an elephant, and they live in a village all together, and an elephant arrives in their village, and so they send the 6 wisest blind people to see what an elephant is made, what it looks like. So they touch this elephant, but each one observes but a small part of the elephant. Now, and you see a particular part. So somebody will touch the leg, will say something, an ear will say something else, the trunk, and so on. Our scientific ability to study plants and their interactions and microorganisms and the environment has gone from the chemist or the biologist or the genetic specialist who was like a blind person to being able to look at an elephant as a whole, and therefore discovering much more carefully what the organism that we're studying is and looks like. Also from a chemical point of view, if we want to have a practical example, in some cases this is changing the way in which we have interpreted some phenomena. I'll give you a case in point. When we talk about wine and the terroir or the soil of the land, we've always taken into consideration the makeup of the soil, but we've also now discovered that the microbiological part of the soil, which can change very much from one area to another, from 1 kilometer to another, influences very strongly the type of molecules that the vine is going to produce, and which we then find in wine, and the type of taste that you have. This means that it's gradually, we are having to change the way in which we will have to impact or describe these questions. Yes, we've had a few questions, very interesting, but I'm not going to read the questions now. I'll read them at the end. Paola Bonfante, we were saying that these microorganisms are useful for the plant, and in particular they're useful for the plant's defense and for its nutrition. Now, in terms of nutrition, could you give me a few more details about that? Yes, I would like to give 2 examples. One is the classical one, that is to say, of those who study plants and biology and agrarian or agricultural studies, because it's very interesting, the symbiosis which exists between bacteria that are called rhizoma, that is to say, they rhizoba, it's with a B. It's a rhizoba or rhizobia or rhizobia, and they are nitrogen fixating bacteria and pulses or leguminous plants. Now, what happens? There is a molecular dialogue. Plants need nitrogen, as we know, so much so that fertilizers usually have phosphorus and potassium. And this is strange because we are surrounded, we live in an ionosphere where nitrogen makes up 80% of our atmosphere. So why do we need nitrogen? The fact is that we cannot use the one in the air, and some microorganisms are able to, and this means they reduce it, transforming it in ammonia, and the plant benefits from it. But it's not so easy. Why? Because what do these bacteria that live in the ground, the lovely soil that we saw before, what can they do? And they are you see them very beautiful and easily recognizable. What do they got to do? Well, first of all, they have to meet, with the plant. So there is a molecular dialogue. We know what molecules the plant releases, how the bacteria answers or responds, and how the plant does it. Because there has to be a mutual recognition, and this is called a molecular dialogue, which leads to a release of these molecules, which in fact are what these rhizobia produce. And this leads to the development of a new organ, which in itself is an extraordinary response. So that is to say, the plant invests a lot of energy to develop this organ. And if you pull up a plant of beans or peas, you see they're pinkish if they're active. And within these structures, you can see the section there, the bacteria enter and multiply and thus fix the atmosphere's nitrogen. And this is extraordinary because if you think that the industrial need for ammonia, it's very expensive, but in the environment, the bacteria know how to do it. Why is it so interesting? And I wanted to show you here a colleague of mine who I admire, Eda Condorosi, and she has received a prize, the Balzan Prize, by the President of the Republic, Mr. Mattarella, because of her study on molecular exchange and communication. Why? Because this opens a new perspective. If we think of those countries where the Green Revolution failed to have a success, think of Africa, where the small farmers have no access to fertilizers. We know that the yield of a maize field is 20% of its actual potential. So the great challenge is to say, can we transfer this symbiotic capacity to plants that are not pulses but grains or cereals? Well, there are a number of projects. A number of step forwards have been made. I'm just mentioning one, which you can see here on the slide, which is they're all funded by the Melinda and Bill Gates Foundation, millions of dollars. The question is, can we see that other plants, that is to say grains, cereals, wheat, will have the same sort of reaction? This would have a social impact, and it gives us an idea of what the implementations of this symbiosis and cooperation processes could do. But if I can give another example, which clearly is very important because I started with mycorrhizae or mycorrhizae, is clearly the fact that we should have a look at the big allies of plants, but maybe we haven't yet defined them. Yes, of course, I will define them in a moment. When we say this, we mean those symbiotic associations that are implemented or which take take place between the roots of the plants and mushrooms that belong, and fungi which belong to a different groups. So it's a sort of umbrella term, let's face it, because it includes a number of different situations. But the basic means of this is that they are in the soil, therefore they colonize the soil, but then they enter the root, as you can see in the drawing, and they establish an interaction which can be very diverse with plants. And I would like to show you here this woodland, which is an imagined woodland that my colleague Andrea Genre has drawn for us, which shows a situation. There's a theoretical woodland, but it could in fact be in the Mediterranean area, where you can see, for example, pine trees and oak trees, and they are colonized by fungi or mushrooms that could be, for example, Russulae or Boletus edulus or Porcini, as they're known here in Italy. And here there's a very long list, and they're ectomycorrhizae, but each plant has its own type. Then there are plants like heather, for example, very beautiful ones, and they have yet another type. And orchids are associated with others. And lastly, all the plants, should I say the more herbaceous ones, are associated with the arbuscular mycorrhizae. Plant clearly releases reduced carbon, that is to say, a product of photosynthesis, which are mainly lipids and not just sugars, and they receive from the mushroom minerals, potassium, phosphorus, nitrogen. So the mushroom behaves as a biofertilizer. But what I would like to show you to mirror what we were talking about more in general, that is to say that the plants are not an island, is that if you look in the boxes below, what do you see? You see that there are networks, underground networks of myceliae that put the plants in communication with another one. And so the mushroom is a sort of repeater, a bridge, putting two plants into communication. We see this with the various types, but there are also situations where mushrooms that, I don't know, have, say, a symbiosis with a pine tree, but can be associated to an orchid. In other words, the network is more complicated. And the idea is that there are not only substances, but also communications that go through. And this is one of the main subjects which explain the importance of the fact that plants are not an island. These images, and where does it end? Where does a plant end? This is a very interesting question. We have an idea that these networks really expand over very large areas. If you can just imagine, there's a colleague, a Canadian colleague, who is specialized in this, Dr. Smart, and she saw that the networks are bigger, and they become bigger when you have what we call a mother plant. So, for example, if you have a tree and then beech tree, and it nourishes all the little trees around it, so there is a physical continuation, or there's an uninterrupted link between the mother plant and the plantlets that have not yet reached independence, because they can't carry out photosynthesis in a good or effective manner because they're too short, for instance, or not yet developed enough. So it's a very interesting question. So I'd like to ask Renato Bruni what a plant is. Well, very different from what we imagined so far. In fact, because of you were talking about the undergrowth, one of the things that we're trying to understand, we'd sort of discovered it a while ago, but we're trying to understand how it works, is, for example, what is an orchid? Orchids have a very particular feature. That is to say, their seed has not enough energy for the new plant to germinate, as happens with many other plants. They haven't got this because orchid seeds germinate only thanks to a symbiosis with microorganisms in the soil, which do exactly what Professor Bonfante was saying, that is, give the nourishment, and when it grows, it will releases part of the investment, nourishing the mushrooms and fungi. So what are we looking when we look at an orchid? Well, we are looking at an orchid, but we're also looking at the microorganism. Or if we talk about a festuna, which we mentioned earlier on, or if we look at plants that interact with mycorrhizae, or are we looking at a plant, or are we looking at a more complex and bigger organism? We sometimes use the definition superorganism to try and convey the idea that what we were looking at in a rather limited way, like the blind people who were trying to understand what the elephant was, is something that we have to understand in a more general way. Likewise, we could say, who are we? Because without microbial foam, I was saying to my students, if I were to become completely sterile, I would not last long. I would die in the next days or weeks. I would die. So am I myself, or am I a series of organisms that cooperate to have a reciprocal help? This is a sort of philosophical approach to life, because as well as competition, we also have to take into consideration these networks or cooperation that we have amongst various organisms that determine the essence of life. I think you agree. Yes, absolutely, I do. And also in the case of orchids, which is a very interesting case that we have studied in Turin. In fact, my colleague Silvia Perotto was also involved in sequencing some of these mushrooms that are very different to the mushrooms that are associated with mushrooms that are with trees. But we've seen that what is extraordinary, how these mushrooms in the course of evolution have learned how to live with the plant. So, for example, in the case of the orchid ones, is sort of borderline. They're there and they're not really sure of what they're doing, so to speak. The others have identified a route in the course of evolution, a route whereby they lost all the genes that might be offensive, that might trigger a defense mechanism of the plant. So, for instance, and it is surprising, there are some that are inside the root, but in fact they have lost all the genes that code for the degradation of the cell wall. This is something really extraordinary. And therefore, how do they communicate? They communicate with a series of molecules that we have studied, and with, for example, with what happened with pulses or leguminous plants, and they release small proteins, which are the ones that are used to communicate with the host. So we think that this is extremely interesting in terms of application to understand how these plants that underlie our nutrition, how do they live with these fungi and mushrooms, and how we can use their tools to the best, since they, as we said, they behave as biofertilizers, but also as bioprotectors. And this is something which is very positive. Right, since we are running short of time now, I think that we could maybe hear some of the questions. Yes, let us start with the first one, because you always start with the first. Now, it comes from Carlo in Turin, who's following us, and I hope that he is following us with others. And the question is the following: how is climate change influencing these microorganisms? What can we expect from these microorganisms? How will they change with climate change? I can say that I have no direct experience on this, but there has been a very interesting review a few months ago on this journal, Nature Microbiology, of a group of experts that highlighted the impact that there is. On the one hand, for example, microorganisms, can they too change or contribute to the climatic change, and vice versa? The climate has an impact on microorganisms, and this means that, for example, our balance is based on a buffer, which is the ocean, and all the microorganisms from cyanobacteria onwards are the ones that regulate through photosynthesis both the fixation of carbon dioxide and all the climate change that affect the cyanobacteria. And on the other hand, it possible to have the sink, that is to say, the accumulation of CO2, and therefore the other important element is linked to all the microorganisms that are in the soil. So we see immediately how both the microorganism in water and the microorganism that is present in the ground, in the soil, have a role which is that of regulating geochemical cycles, are key, and therefore definitely in relation to climate change. But I would say that the oceans is certainly the most important impact. This other question comes from Trieste, so thank you very much for following us from Trieste. And Sylvia asks us the question. It's a very interesting question, and it's also very topical. She was saying in terms of the situation that we have, COVID-19, that is to say, we are talking about the fact that the virus jumped from animals to human beings, but could it also happen from plants to human beings? I'm going to take time, and I'm going to add something to the answer to the previous question. First, you were saying that on the surface of leaves, there are microorganisms that have to find nutritional substances that are not available. Many also use methane to do this, and methane is one of the substances that we're trying to reduce in the environment with the problem of global warming and so on, is leading us to lead it, because it's a sort of greenhouse effect, yes. And then today we spoke a lot about balance or equilibria. Well, clearly the climate change introduces a change in the balance, and what was stabilized becomes needs to find a new balance or a new equilibrium. Many of the microorganisms that I mentioned, I spoke about hosts, live in a balance with the plant, so long as the plant is able to cope with the balance. If, due to the climate change, plants encounter more difficulties, some of their inner endophytes might no longer be able to be controlled as guests, but might become more difficult. And therefore, rather than being a benefit, they might hinder the life of the plant. As far as the species jump or leap, well, there are plant viroses, but it is also true that the plant cell is very different from an animal cell. And so the many viruses that affect even cultivated plants, they will not necessarily recognized or be recognized and therefore have an aggressive dynamic. There's a different basis between plants and animals, so it's rather difficult for there to be a leap from a plant pathogen, unlike what happens with animal to animal. Yes, it is something, without talking only about viruses, because I haven't got many ideas on viruses, but talking about bacteria that are part of the plant microbiota and which then would develop in our body is difficult because of the differences that I was showing before, that very often they're aerobic and very also react to our environment, and our gut is acid, so it's not so obvious, at least for bacteria, that they could go from one to the other, from one microbiota to the other. But in fact, they found also, and I was very surprised, they found sequences of arbuscular mycorrhizae mushrooms in the gut of the microbiota in animals, or I think that was probably, it was dead DNA that had survived as a trace of food or something, because I could rule out that it can live in our gut. Alessandro from Turin asks, do we know how important the so-called green manure system or cover cultures are? I would like to know whether for microorganism could this enhance the effect? It's quite a technical question. Yes. Well, the cover effect. Did you say it? Well, yes. They're cover crops. That is to say, they're plants that are put in the winter time, for instance, or when the soil might remain bare. This is quite an important one, especially in the Po Valley, because the fact that you have kept the fields bare leads to a loss of nutrients from the soil and a washout and an impoverishment of the soil, and the fact that the new plants find a poorer soil. But this is partly compensated, offset, by the idea of putting cover crops on and that would cover the soil in the time. Surely there are elements that favor this interaction, because these plants make it possible to have a continuity for the microbial population, and I'm talking about yearly plants clearly. I'm talking about them, the fact that the crops can use more microorganism, or these type of interactions are favorable. There are also, in some poor soils, you have lower yields, worse crops. In fact, one of the techniques that is very simple, but which is still advised for, is the so-called green manure. That is to say, to alternate a pulse with a grain or cereal. Thinking about Africa too, because this automatically enriches the nitrogen content in the soil. And then there's a number of techniques that are called agroecology. That is to say, you take a natural situation where, for example, there are several plants in an environment, and then use them in agriculture, obtaining crops with different plants which have different microbial communities and which enrich the land. So it was a very appropriate question, a very interesting one. We have several questions, so let us see if we can answer them quickly. One of them is, again, by Alessandro in Turin. What is the role of microbiota in the use of water in arid and semi-arid areas?
Posso dire qualcosa? Allora, sì.
Yes, there are a series of very recent publications that focus on Indian millet or sorghum. That is to say, it's a very interesting plant because genetically it is more resistant to drought than, say, sweet corn or maize. So at this particular moment, there is a focus to say that we could try and develop these plants and see how the microbiota works. As we have already said, that the microbiota changes according to the conditions and availability of water, but the thing that could be a little bit worrying for us, that is to say that where there is drought, what do we see? We see that there is a reduction of the benefic effect or the beneficial effect with the arbuscular mycorrhizae. So they too seem to suffer of this state of drought, or while this is the experiments in the field, while in the greenhouse it looks as if it would help. So to give an answer, we could say that surely there is a relationship. Studies are underway, but unfortunately the response is different in greenhouse or in the field, and so we have certainly gain a better understanding of the identification of targets. An environmentalist concern: when you standardize agriculture with chemical products, pesticides, herbicides, monoculture, are we not limiting or influencing this biodiversity? Are we not artificially selecting the microbiota in this area? By Paola. The question is by Paola in Turin. Yes, we're discovering that some things are more complex than what we thought. So clearly, when we didn't know that microorganisms were so actively involved in the life of plants, we worked without knowing that, without having that information. Now we know that these things are very important in terms of the crop yield too. The important thing is that knowing that this, which is physiologically part of science, that is to say that progressively we discover new things and we have to bear them in mind in developing activity, that we should have an approach that we say is evidence-based, based on the fact that we should have a non-ideological approach. So when we talk about agroecology, we're talking about bringing these suggestions and implementing them in farming. For example, the green manure cover crop is again this sort of thing. These studies that have been carried out to try and convert some of the crops, for example, having rather than yearly plants into other plants so that the land will be more fertile. Whatever action we do has an impact.
Questo non riduca magari eccessivamente le rese.
Certo, se posso aggiungere, noi sappiamo che sia microorganismi, questi plant growth promoting.
If I can add something, we know that both microorganisms, and then these plant growth promoting bacteria and the mycorrhizae are already part of inocula that are sold by several companies. So the inoculum itself is sold in Europe and is applied on the fields, say in France, just as in cassava in Colombia. So the question of the listener is absolutely correct. Is there What is the meaning? Are we influencing in some areas the outcome by transferring a non-endogenous microbiota, which is linked to what we were saying before, that the same plant in different environments behave in a different way? Yes, it behaves in a different way. And also in the past few years, we've had agricultural practices that have not favor the growth of the microorganism. And now that we've discovered it, we have to understand what is the best way to regulate our agricultural policies and not lose the benefits that they have. Yes, there's a very brief question for Renato Bruni. Why did you call plants bad animals? Well, it was something my grandfather used to say. When I spent time with him in the green garden, when I was not at school in the afternoon, I used to work with him in the green garden, and he used to say it because plants did not do what he wanted them to do. And so he would say there are bad animals and that they do what they're interested in. We would like to force them, but they do exactly what they want. There is a question by Sandra in Turin, but they were showing me, in fact, has sent an additional pointer to the smartphone. She said, in any case, the question was the fact that some microorganisms cannot be cultivated. Does this mean that they're not autonomous? In other words, are there cases when the symbiosis? Well, here, this is the additional. Please, the question was not well clear. Is there a necessary link with the host genome? This is quite a sophisticated question. There can be different types of reasons why they can't be cultivated. For example, a microorganism can have in its genome all the potential metabolic routes which are required to grow, and we don't know how to recreate the right substratum, while there's another group of microorganisms that cannot be grown or cultivated, where the genome has been reduced, and this is the classical example that we find in the microorganism that we call endobacteria, both the endobacteria, the ones of the symbionts, mushrooms or fungi, but all the ones of insects. In this case, these bacteria have very small genomes, and therefore they're not able to do certain things. For example, they cannot create or build certain amino acids, so there must be a functional existence with the host. So she's absolutely right. One of the reasons why we only recently discovered them, because we didn't know how to cultivate or grow them. We were not able to do so because we didn't know the ideal conditions in which they would thrive. Alessandra, who writes or phones from Candiolo, sent us the message. Due to following the fires in Australia a few months ago, billions of microorganisms must have died. What is the impact for humans? Who wants to answer? For the Australians, maybe. Yes. Well, they too are human. Well, in terms of climate change, there will be macro consequences for us too. Yes, part of the microorganisms in the soil may be very deep down, and so there is a reserve which progressively will repopulate the ground closer to the surface. Microorganisms are very flexible from this point of view. Think of the difficulty that we have to eliminate the pathogens that we have. We've been trying for years, and they reorganize and rebalance. So the existence of a small population on the surface for example, of plants that were not burnt down will surely repopulate the others. A last question, which is very interesting and also looks into the future. Camilla from Turin, on the INAF, that is to say the National Institutes of Astrophysics website, and you see there's a harmony with from stars back to stars. On the INAF website, I read about a study which concerns the role of mycorrhizae in space gardens. That is to say, the green gardens or the cultivations that they're trying to do in International Space Station, or in the event, for example, of a more distant space station. There is one which is being planned around the moon, which should be implemented towards the end of the 2020s. So can you tell me something about the possibility of these space green gardens? Honestly, I have no direct experience. Several times in the past, we had spoken about these projects. That is to say, the use of the mycorrhizae to inoculate these plants so that they might support or help creating an environment which is very difficult. Now, it can be something very useful. What I see is, and I'm going to just say it as an interesting solution, albeit a non-spatial one or non-space one, that is to say vertical gardens. Vertical gardens are really a possible implementation because they are plants under stress. And so certainly, as we say that our fungi and mushrooms alleviate, or fungi alleviate the stress, I think we could probably say that this happens in space gardens, but I don't know what the results are. But I will go and check out the INAF website. So thank you very much. I finished questions. Oh no, there's still one question. Can a weak flora in our gut have an impact on our lungs and heart? Turin. Well, our gut flora is part of the immune system. I don't know if it affects the lungs and bronchi, but as we were saying at the beginning, it is one of the main functions of the microorganisms is to take part in our immunological system. So the answer is probably yes, but a weak or altered gut is bad for everything, whatever impact it has specifically on lungs. But now, going back to plants, I would like to give the floor to Paola Bonfante if she has something to say. And if she can conclude, yes, I think that I would just like to show my last slide, which summarizes this evening's talk. That is to say, we think that to study and better understand these interactions of fungi, mushrooms, bacteria, endophytes is really essential or key for the health of plants, for the environment, for agriculture. And I think that this potential revolution will require a lot. But I feel that our ideas are a bit clearer, of a sort of triangle where we have plants, human beings, the environment, and we say if the plant is healthy, if human beings are healthy, if the environment is healthy, we then have a single notion of health, and in these three conditions the microbiota is really a common keyword. And I think that this was extremely interesting to introduce this, because normally people say microbiota, wonder what that is, but it is absolutely present and important. Thank you very much.
Grazie, arrivederci a tutti gli amici dello streaming.